Okay, next we're going to take a look at the inflammatory response. And just to back up here real quick, inflammation is another part of the second line of defense, the innate or non-specific immunity. So really inflammation, and then we're going to talk about fever as well on these next two parts here. Now, inflammation is a very broad topic, and it gets into a lot of different things. It's described in our textbook anyway as at least a response to a reaction to any traumatic event in the tissues. And so inflammation is something that we often just think of as swelling, redness, maybe pain, and that usually can happen due to a variety of reasons. So I should say it can typically happen due to a variety of reasons. So for example, a bruise is kind of like a type of inflammation. So if you hit yourself or something hits you or you fall down, it could happen if you get an infection. Maybe you have an infected hair follicle. Maybe you have an injury to yourself. You poke yourself with something. Maybe you burn yourself, or you have a chemical burn. Rashes. All these things are a type of inflammation, and so really it manifests in lots of different ways. So it's a very broad topic. So what we're going to do is we're going to look at it from a very specific type of angle. We're going to look at it from what we call an acute angle, or what's called acute inflammation, that is, rather. So inflammation, you can have acute inflammation, you can have chronic inflammation. Inflammation can be local, meaning it's refined or restricted rather to a certain area, or it can be systemic, traveling throughout the entire body or occurring throughout the entire body. And all of these can really be discussed in a different context. So chronic inflammation is really quite different from acute inflammation, much less obvious. Systemic inflammation is going to take on different characteristics than local inflammation. So rather than try and break down the whole thing, we're going to look at it from an acute local inflammation standpoint. So in that sense, inflammation often is more specifically characterized by redness, warmth, swelling, and pain. And all these things can also take place on a systemic level, even on a chronic level, but not always to the same degree. And it's not always this extreme here, like you see in this picture, but oftentimes it's some degree of this. Now, all of these really are characterized or are caused by increased blood flow to the area. And really that's what inflammation is all about. So redness, the color, is actually given off by increased concentrations of red blood cells. Same as the warmth. You have more blood flowing to the area. Blood carries warmth, and so the tissue gets slightly warmer. Swelling is caused by the fluid itself. Fluid is literally leaking out of the tissue or leaking out of the blood vessels rather into the tissue. And pain can sometimes be caused by the pressure of the excess fluid pushing on nerves in the area. And occasionally, if that happens to an extreme degree, it can cause decreased function. Like if you, for example, hit your thumb with a hammer, and then your thumb can't bend anymore because it's too swollen, although that's not as common. So these are all really a characteristic that results from increased blood flow to the area. And so as I mentioned, it can happen for a variety of reasons. Trauma from infection, injury, even one I didn't mention would be a certain type of immune reaction. And we'll talk about this in chapter 16 actually, but an example is like a type 3 hypersensitivity reaction. So these are a variety of different things that can cause it, some more common here. So we're really going to look at it in the next slide here, next couple slides, in response to really kind of an injury and infection simultaneously. Now, before we look at that, the functions of inflammation really serve kind of three general roles. And again, this is more of an acute local inflammation, really more of from an acute standpoint in general, I should say. And what's happening is the body is really signaling an alarm to some trauma in the body. And that alarm is ultimately geared towards attracting and mobilizing immune components. These include white blood cells, first and foremost. And the different signals that they give off bring in even more aspects of the immune system, like antibodies and things like that. So ultimately, it's an alarm system saying there's something happening here. We need to get some help over here, and so that is part of that process. I put these a little bit out of order. Once the immune cells get there, white blood cells, they're ultimately trying to destroy any microbes and prevent further invasion throughout the body. So they're not only going to destroy microbes, but they're going to kind of set in motion this kind of process that's going to wall them off and prevent them from spreading, sometimes referred to as granulomas. And then ultimately, they're going to set in motion repair mechanisms that allow the tissue itself to recover and reform, regenerate, and ultimately try and put itself back into as close to form as it was prior. So it looks something like this. And so this is again an example of where you have really an injury followed by infection at the same time. So this is some kind of needle or some kind of nail, perhaps. So it's piercing the tissue. So here's your skin, the nail or whatever this is piercing the tissue. And when that first happens, you've got injury, and the body is going to respond in kind of a knee-jerk fashion. And what's happening here is when this piercing event takes place, cells that line the tissue, these cells here are called mast cells, they're going to immediately release chemicals that are going to cause a response in that area. So it's very much like a knee-jerk reaction. And one of the first things that's going to happen is that the vessels in that area are going to constrict. The blood vessels are going to constrict, and we call that vasoconstriction. So it's a narrowing of the blood vessels. What that's going to do is it's going to kind of cause the tissue to kind of prevent immediate leakage. So in the very beginning, it's trying to kind of prevent fluid loss, and then that's actually going to change. It's actually then going to increase fluid loss shortly thereafter. The idea is that this kind of helps some of the clotting factors initiate, and it helps the body kind of start that repair mechanism by preventing excess blood loss. So you want to allow the wound to clot, and then the tissue will actually start to swell thereafter, which will then allow some of the cells, the immune cells, to come in. So the constriction kind of helps prevent excess blood loss. And so that's what you see here in this next stage. After the constriction has taken place long enough, and theoretically a blood clot will form that prevents excess fluid from being lost, excess blood from being lost, that is. And the next thing that's going to happen is now the vessels are now going to dilate. So you go from a constriction to a dilation. With dilation, what's happening is now you're going to have fluid that's going to leak out into the tissue area. From this figure, it looks like all the fluid would come through this opening here, and some of it certainly would. But even in areas outside the immediate wound here, and this is going to get clotted to a certain extent, keep in mind, what's going to happen is that the vessels are actually going to start to leak. So here's a figure off the web showing what this might look like. Under normal circumstances, the cells that line the blood, that make up the blood vessels, they're relatively tight, and they form a relatively waterproof layer, not waterproof, but a relatively watertight seal. But the vessels themselves are not exactly like a hose. They're not watertight. They do have an ability to leak, and that ability to leak can be exacerbated during vasodilation. So it looks like this. You've got the vessels that all connect the cells, that all connect the vessels are somewhat locked together, almost like puzzle pieces, right? And then when vasodilation occurs, those puzzle pieces kind of loosen up, and you get these kind of gaps in between the cells. And that allows some of the fluid to literally leak out into the surrounding area there. So ultimately then, that is a big part of how fluid gets into the tissue. And then also, as we're seeing here, some of the white blood cells are actually going to crawl out through those holes and migrate into the area as well. And going back here just real quick to connect this, what we said earlier, this is part of how the monocytes differentiate into macrophages, and they're actually going to crawl out of the blood vessels and into that tissue. And you can see here, once again, they're showing you kind of connected areas here that they would expand, and those would actually be able to crawl out. So ultimately then, we go from a constriction for clotting to a dilation to fluid seepage and accumulation, as well as cell accumulation in the area. So now things like neutrophils and the macrophages in short time would start to seep in. I misspoke there. These would be mostly neutrophils migrating into the area first. Ultimately then, the neutrophils are going to start to kill off a lot of these microbes, and the accumulation of pus is going to start to take place. Now, this is going to take place over a certain number of hours, probably even days. So, as the neutrophils start to kill off these microbes, pus starts to form. And pus really is a combination of white blood cells and phagocyte debris. It's kind of like a battlefield. It's like the remains of a battlefield left behind. So ultimately here you've got the body kind of fighting off infection beneath the surface here. Also, the body is laying down some of this fibrous tissue here, and that's theoretically going to help prevent microbes from getting spread throughout the body. So it's kind of trying to trap them in that area, prevent them from migrating further. And then ultimately, the body should theoretically handle these microbes, should handle the infection, and that should lead to scar formation. And as you can see towards the end, macrophages will come in. They'll scoop up, phagocytize lots of these microbes, and then travel back to the lymph nodes in the spleen, where they will trigger a third line of the immune response. This will allow the body to recognize these same microbes later and provide a more advanced immune response involving antibodies. Or if the infection is still not cleared, if this is becoming a long-term infection that's not being handled properly, the macrophages will go trigger a T and B cell response that can migrate back into the tissue and ultimately cause a more advanced response. Usually in this kind of scenario that doesn't typically happen, but it can. But in a kind of an average scenario, you would have clotting followed by fluid accumulation, which leads to pus, and then ultimately a resolution. And that's kind of a general sense of what acute inflammation would look like, acute local inflammation. So again, ultimately, set in motion repair, bring white blood cells, destroy the microbes, and set in motion repair mechanism. Now, part of the big thing that's happening here are vascular changes, and these are really some of the things that are key to inflammation. And when you have a puncturing event or a bruising event or a trauma event, you're going to stimulate nerves and as well as some of the tissue there in the blood vessels themselves, and those are going to release chemicals that are going to trigger some of the vascular changes themselves. So if we're looking back here, I showed some of these mast cells, for example. They release chemicals, and that actually is what leads to vasoconstriction, and then ultimately different chemicals lead to vasodilation. So any kind of chemical that affects the blood cells, whether in a constrictive or dilated way, these are considered vasoactive mediators. Other chemicals that are released have an effect on the white blood cells themselves. These are called chemotactic factors, sometimes called chemokines, and they're the chemicals that will actually draw in some of the white blood cells, like macrophages. Now, some of the terms here that relate to how macrophages and phagocytes are changing their behavior. One of them is called diapedesis, and then chemotaxis. These actually kind of go hand in hand, so we'll talk about both of them. Remember, chemotaxis we described as being a response in a chemical movement, in response to chemicals. And so what this is again applying here. So the white blood cells are chemically attracted, in this case, mostly to chemicals given off at the site of the injury or infection. So they could be PAMPs, the pathogen-associated molecular patterns that we said earlier, but probably more prominently in this case, they are chemicals given off by your own cells as a response to a trauma. So, for example, some of the mast cells releasing chemicals that would ultimately attract them in. Neutrophils release chemicals that attract macrophages. So chemotaxis is kind of, in this case, a general term in which these white blood cells are migrating towards the traumatic event. And diapedesis is a term that describes how they literally crawl through the vessel lining here in your blood vessels. So the vessels, I mentioned, are kind of connected like puzzle pieces here. And as they dilate, these white blood cells can literally crawl through them, travel out of the vessels and into the tissue around them. And that's ultimately how they get from the vessels into the tissue. So that's called diapedesis. Okay, so that's a quick summary of acute inflammation there. Next, I'm going to talk about fever just shortly here. Fever, as we know, is an increase in body temperature. We've all probably experienced this at some point. Fever, though, is actually something that is controlled by a part of the brain called the hypothalamus. Hypothalamus does many things, so it's not just what controls fever. It's a very complex endocrine part of the endocrine system in our brain, and controls many different aspects of our hormonal system in the body. So a very complicated part of the brain. But one of the many things that it does is it plays a role in fever. And fever ultimately is a response to molecules called pyrogens. And a pyrogen is a fever-inducing molecule. The term pyro relates to fire and heat. So think pyro, think fire, think pyrogen, fever. And ultimately what they're doing is they're causing the hypothalamus to reset itself to a higher temperature. So you can really almost think of in the hypothalamus in this context is almost like a thermostat on the wall that controls the room temperature. And a pyrogen is basically the equivalent of someone pushing the button and increasing the temperature. And ultimately that causes the body temperature to rise. Now pyrogens can typically be of several different sources. Many of them will occur inside the body. These are called endogenous. They originate inside the body. Most often these are produced by phagocytes. So phagocytes are obviously the white blood cells that phagocyte, and anyway they produce chemicals during that phagocytic process. And there's a threshold here. They don't do it right off the bat. So this is a more in-depth part of the immune system, but there's kind of this delayed effect where if the phagocytes are active long enough and there's enough of them and they reach kind of a quorum, then they will eventually start to release pyrogen. So that's not really a knee-jerk thing, but more of a delayed, kind of a long-term in response to a more traumatic type of infection. This is why you don't get a fever every time you have a little minor hair infection or like a hair follicle infection or something like that. So they're not just going to automatically trigger fever right off the bat. But that can ultimately take place due to phagocytes. Occasionally, you'll be exposed to chemicals, pyrogens, from an outside source. These are called exogenous, and these are the different kind of different class of pyrogens that can ultimately do the same thing, but they come from an outside source. So some examples of these would include some of the products of things like viruses, bacteria, protozoa, and even fungi. So some of these types of pathogens can actually give off chemicals that ultimately cause your body to go into a feverish state. One example is endotoxin. Endotoxin comes from the lipopolysaccharide I mentioned as being a type of PAMP. I mentioned that being part of the gram-negative outer membrane. So gram-negative bacteria are particularly known to induce fever given a relatively serious infection. Now, once again, they're not going to do it just automatically. There are kind of threshold levels here, but they are able to cause a fever if they have a serious enough infection. Some species are more potent at that. For example, Salmonella typhi is a microbe that causes typhoid fever. Typhoid fever is a name given to the fact that that particular infection is well known to cause a high fever. Other things like vaccines, injectable solutions, even certain types of blood products, like if you have an inappropriate blood transfusion, these things can ultimately cause a pyrogenic response. Endogenous, as I mentioned, often come from phagocytes like monocytes, neutrophils, and macrophages, and can ultimately trigger fever due to a kind of a natural immune response. That's most typically how that's going to take place. These are typically a natural response to infection. Now, one of the reasons why the body wants to do this in the first place, because fever, as you may be well aware, fever can be a real problem. If the fever gets too bad, then it could actually be detrimental, even fatal in some cases. Our body has kind of a sensitivity to too high of a temperature, obviously, and sometimes the fever can be very detrimental. But ultimately, the idea behind the fever is that it does have benefit at the right level. So if it's not too high, there is a benefit to it. And one of the benefits is that certain microbes are temperature sensitive. And if the temperature gets too hot, they just simply can't replicate, they can't function properly, and that can be an advantage to the body. Kind of either slows them down or prevents them from replicating. So certain types of cold viruses, like a rhinovirus, for example, is really not able to replicate at a high temperature. So if the body's fever goes up, the microbe replication goes down. When it comes to certain bacteria, bloodborne bacteria like streptococci and staph, the ability for them to absorb iron goes down as body temperature goes up. So this is a kind of an enzyme substrate complex interaction that is reduced due to a higher temperature. So a slight amount of denaturation of the enzyme occurs, and iron absorption goes down due to that denaturation. So it's a sensitive enzyme. So the idea is that iron is often a limited nutrient, and it kind of limits the rate of their growth. So microbes can only grow as fast as the least amount of nutrients that they have. So they may have lots of carbon, they have lots of hydrogen, but iron is necessary for growth, and it's usually the most difficult to get. So by slowing the rate of that particular nutrient down, it slows their overall rate of growth down as well. So that can help the body gain a foothold on those microbes. And ultimately, it's thought to increase the metabolism and stimulate kind of a general increase of immune response throughout the body. When things heat up to a certain degree, they tend to move faster. And so as the body heats up, theoretically things move a little faster, the immune system speeds up, and circulation speeds up, and things are kind of taking place at a slightly faster speed. Again, this all has thresholds, at about 100 and 101, maybe 102 degrees. There may be a lot of benefit to that. You get up to 105, anything past that, and suddenly you're looking at some pretty serious consequences. So we're all within reasonable limits here. So that's kind of the benefit of fever and kind of how fever works here. The last little bit here is taking a look at a couple examples, go all the way back here, of antimicrobial proteins. So we've talked about phagocytosis, inflammation, fever, and one of the last examples of innate immunity are antimicrobial proteins. So these two that topic in general, antimicrobial proteins, can really get complicated. So I'm going to really try and keep this simple here and not go into a lot of detail. So if you look at your study guide, I just have a couple questions about the two examples I'm using here. So nothing really in depth. So I'll try and keep this last little bit short here. But interferon is a really interesting process that takes place in response to viruses, typically. And when a host cell rather is infected by a virus, one of the things that our cells have evolved to do is to literally send out a signal to other cells in the nearby area that they have been infected, or that cell has been affected. And what happens here is the cell releases a chemical that will bind to the nearby host cells, and that activates internal cell pathways that are typically effective at breaking down viral DNA. So when a DNA virus comes into the body, it will typically infect a certain number of cells depending on how many virus particles you're exposed to. And each of the initial cells that are infected will put out an interferon signal, which will then trigger other cells in the neighborhood, nearby tissue, that will give off that will activate a pathway that will prevent them from being infected at the same rate. So it's a lot like a warning signal. It's almost like if you have in a neighborhood, if you have someone going around breaking into houses or breaking into cars, one house gets broken into, maybe two houses get broken into, and they're going to signal to the rest of the neighborhood, hey, we've got someone going around breaking into houses here. And everybody in that neighborhood's now going to be on alert, right? Especially in Kansas, where everybody's got a gun. That criminal is probably not breaking into too many more houses successfully. So it's kind of like that. You've got a small number of incidences that lead to a warning to everybody else to be on alert, and that makes it much more difficult for, in this case, the virus to be effective at continuing to infect. So it looks kind of like this. A virus comes in, this first cell is infected, but what it does is that triggers a synthesis of this interferon chemical. And that interferon is released and binds to other cells in the neighbor area, and that changes their response and ultimately activates pathways that otherwise wouldn't be active. And those pathways ultimately will break down incoming viruses as they attempt to go on and infect those other cells. Once these pathways are active and the virus tries to infect them, it breaks down the virus after it comes inside the cell. So this is kind of an elaborate evolutionary mechanism to viral infection, and this is one of the reasons why our body is less susceptible, I should say, to massive infection by virus. If you mentioned from chapter 6, we're talking about viruses. One virus can potentially make hundreds of thousands, if not millions, of copies of itself in just one cell. And then each one of those could theoretically do it again. So if you do the math, one virus could easily lead to 1 million more viruses, and each one of those viruses could lead to 1 million more, and so the numbers would be overwhelming relatively quick. One of the things that stops that is the fact that our cells put out signals that often prevent them from continually infecting every cell thereafter. Here's another video. I'll post a download for that online also. And the last example here is another type of antimicrobial protein. These are called complement proteins. Complement proteins work in conjunction with the third line of the immune system. So they actually work in conjunction with antibodies, which are produced by the third line of the immune system. So these typically take place is kind of a combination between the specific and the non-specific. So they're technically part of the non-specific. And the reason for this is because they will typically bind to any type of antibody on the surface of a cell. So they don't necessarily have to recognize a certain type of antibody, so kind of any general antibody. So the antibodies are specific, but the complement proteins are not. They just pretty much recognize any kind of antibody. And what happens here is antibodies will tag or coat a cell, like a bacteria. And so that kind of marks these cells, and that indicates that they need to be eliminated. So these complement proteins will then recognize those, and they will bind to the antibodies. And what happens is that the binding of these complement proteins ultimately sets off a cascade of reactions. One protein binds, another protein binds to it, and it follows in suit in a cascade reaction. And the binding of all these different antibodies ultimately triggers a perforation in the cell membrane of the microbe. And so what it ultimately leads to are these puncture sites in the hole in this cell of a microbe. So this is an interesting figure here. It's actually showing the surface of a microbe. And each one of these little holes, like a bunch of craters in the surface, each one of these are actually a perforation by these complement proteins. So the antibodies tag the microbe, and then these complement proteins come in and literally start to align themselves in a way that punctures holes in the cell. Now, the chemistry behind this is actually fairly complex, and this is one of those areas I just kind of choose to not make you learn and get into all the details of that, because it's something that you can spend quite a bit of time actually learning exactly how this all works. And there's all these complicated mechanisms by which the first protein and then the second, third, and fourth proteins all bind. It gets a little tedious. So rather than get into all that detail, we'll kind of talk about the general process. So it's a protein in the blood, the protein series of them, that bind to antibodies. And when they bind, they ultimately will lead to an arrangement that perforates a hole in the cell. So it's almost like this little pattern that aligns itself on the surface of the membrane, and then there's a process that literally punctures a hole after these proteins line up in just the right mechanism. Pretty interesting stuff, actually. But ultimately, the end thing here is that it punctures holes in the membrane, and that causes lysis, popping or rupturing of the cell, pathogen, and death. So this takes place after you've been exposed to a microbe, but it's considered non-specific because these complement proteins will typically bind to just any kind of antibody here that has tagged a microbe. Okay, so that is the end of chapter 14. Make sure you take a look at the textbook questions if you haven't done that already. As always, let me know if you have any questions, and I will get back to you as soon as possible.